everyone. I'm Peri Bolton. I'm a PhD student at Macquarie University. Simon Griffith is my primary supervisor, and I have a co-supervisor down at Deakin doing the genetic stuff with me. Normally this is a two-part talk, but because it's a double act today, I'm going to cut out the genetic incompatibility part, which I believe Simon has talked to you guys about already. Sarah Pryke is responsible for a lot of that stuff. I'm furthering that a little bit, but I'm mostly going to talk about the conservation genetics today. So, give you a bit of a background on where I came from and sort of what conservation genetics is and what does that entail and why I'm interested in doing it, and then I'll talk a bit about my particular research. So I did my undergrad at ANU and I started out as a herpetologist rather than an ornithologist. I used genetics to sort out the taxonomy of these cute little elapid snakes. Still working on getting it published, but you know, no one's ever done this stuff before, and they're really cute, and my heart is in reptiles. I did some volunteering for a while. I did some reptile stuff, and I did some bird stuff. I had the privilege to go to China and work on cuckoo and host coevolution, and how the hosts can respond to the pressure of these birds laying eggs in their own nests, so how they perceive different colours in their nests, and what sorts of things do they do to avoid having cuckoos. And that's a little cuckoo chick in its tiny little nest. So that's kind of cool. Yeah, and so all of that kind of led me to decide that I wanted to do a PhD. And so I'm doing conservation genetics. And I started a couple of years ago. This is my final year, third year. Yes, so conservation genetics is looking at the genetic properties of populations and how that can be used to inform conservation. So for a long time we've understood how the genetic properties of populations, how we expect them to evolve, how, you know, if we survey the population, what they're going to do. And so conservation genetics is a slightly newer version of that. It's just using these principles in a way that allows us to say, okay, well, if we've got this many individuals, what are the genetic properties? What do we predict? And so on. And I'll explain a bit more about that as we go. So it basically is a way of measuring the genetic health of a species. So one metric that we're interested in is population size. So this can be quite different from the census size, and this is one of the main focuses of my PhD, is that the population size as measured by the genetics can actually be quite different from the census size. So the population size as measured by genetics is kind of the number of breeding individuals. Not exactly, but it's close enough. And so, because they are the individuals that are passing on their genetic material to the next generation, so we call this genetic effective size. So this is something that we can measure using genetic tools, is how many effective individuals are there in the population. So there could be a lot of unaffected individuals in the population.
Do you mean by unaffected ones that aren't brother and sister, that kind of thing? The genetics too close?
That is sort of part of it, but it's sort of a more complicated thing than that. But it's helpful to think of it as number of breeders. So the genetic health is also these small population sizes can be bad, which is why we're interested in conserving them. So we all know that small population sizes can lead to inbreeding, and inbreeding can also happen without small population size. But Tasmania, yes. And we all know that inbreeding has negative effects. So in humans, there's all sorts of retardation. So basically, what happens is that it brings lethal alleles that everyone has in the population together, and they're more likely to become surfaced. So you get all sorts of nasty things. So that can have fitness effects. So it means that often they aren't able to breed very well, and they're just they may not be able to find food as easily. There's all sorts of negative consequences. So obviously we don't want our populations to be inbred.
Excuse me, what do you mean by inbreeding?
I mean, that's a good question. Tasmania, again, I mean, so inbreeding depression is what we're worried about. So inbreeding may not be a problem in itself, depending on how many bad things are floating around in the population.
What do you mean by inbreeding?
How close?
How closely related individuals.
Because most species of animals that we have now are the result of line breeding.
Yeah.
Is that what you call inbreeding?
No.
Yes.
Yes.
Okay.
But I mean, it's not necessarily always a bad thing. So it's inbreeding depression that we're worried about, but inbreeding in general in wild populations can also be bad. And we use that as an indication that in wild populations, actually there could be inbreeding depression. So yeah, inbreeding in general, like, is not necessarily a bad thing, because you guys do it in a controlled way. You say, okay, we know this guy has this and this guy has this, and we put them together. That may be inbreeding, but that doesn't necessarily mean it's a bad thing. But we're just using it as a course measure to go out in a population and say, okay, well, these individuals seem to be more closely related than they would be if it was a normal randomly mated population, say, you know, humans or whatever, going out and doing whatever they want. So these guys are more closely related, making babies, and just statistically, that is more likely to be uncovering nasties in the population that lead to all of these bad effects, which is the things that we're worried about. So another thing that happens with small population size is that we get a loss of genetic diversity. So if you think about, you have a small population and you're randomly sampling this population again and again and again, and these different variants, maybe they're, I don't know, resistant to 2 different strains of a disease or something like that. And so as we go through the generations, this random sampling effect essentially means that by the end of it, we've lost one of our variants. And so if we're trying to conserve a species against all possible futures, we really want to be conserving all the different variants in that population to allow it to adapt to the future, and, you know, so that it's not inbreeding as well. So that's another thing that we want to look out for with small population sizes, about how these populations are actually going to respond genetically. And so also another something that conservation genetics is interested in is looking at the connection between habitats. So we all live in a fragmented landscape, and for some animals, so for example this little rodent here, it might live in these foresty patches. But these fields may not be very far apart, but that actually could be a significant barrier. So what actually to us might seem like one big population, this could actually be one tiny population here and one tiny population here. And so how we manage that genetically is quite different as opposed to one whole big population that can all interbreed. And so it might actually be that we have a number of very, very small populations that are quite inbred and have all sorts of nasty things going on rather than one big one. And so looking at the connectedness between regions is also something that we're quite interested in as conservation geneticists. So conservation genetics is widely used in management of endangered species, and these are just some of the species that are, you know, currently being used, or currently conservation genetics is being used on to help conserve them and manage them properly. And Stanley's got a good example that he'll go into a bit more. So, onto my work. So the Gouldian finch, I'm sure you all know this, but I've got to say it anyway. It's distributed in the sort of savanna habitats of northern Australia. And it was formerly distributed, you know, quite widely over this habitat. But over the course of the 20th century, it has become quite a fragmented range. And it's become a lot rarer, mostly due to the effects of overgrazing and fire, you know, the colonisation of the north that was really, really pushed after the Second World War. So yeah, so now it's a very small and fragmented range. And until recently, it was listed as endangered by the IUCN and is currently still federally listed. but the IUCN is like the global body that kind of guides global conservation policy. So if it's been delisted by the IUCN, it's kind of going to want to wonder why. And this has been on the basis of these dry season counts. Now, these dry season counts are very good. They're very useful for a number of different things, but they may not be necessarily very useful for estimating the population size and how connected they are. So these are done in a so they might be systematic within a particular region, but what we're concerned about is that we have a number of non-breeding individuals in that population, so it doesn't represent the breeding population size, and that bird watchers that are observing birds in the dry season in the Northern Territory might actually be seeing the same flocks of birds in Western Australia, because there's no concerted effort across this range here to actually do a real survey. It's mostly based on anecdotes and a few years maybe of surveys, but like it's not a real concerted effort. So using genetic approaches, we can get at some of these questions. So just to outline sort of the three main components that I'll be looking at with Gouldian finch that no one's really looked at before is how many populations of the Gouldian finch are there? So, I mean, on this picture here, we can see that, I mean, this isn't necessarily the most accurate map, but we can see that there are sort of fragments that we might predict that these form distinct populations from each other. Or is it one big population? How connected are these populations? So maybe they are completely isolated and there's no interchange between any of these populations. Or maybe they are quite connected. We don't know. This is something that I'm looking at. And how big is this population? So this is this question of how many breeding individuals there are actually in the population. So I didn't actually get to do any field work as part of my PhD. I had the, some say, luxury of having all my samples collected for me. So Sarah Pryke and James Brazel both worked in the field in Wyndham, establishing a field population there, doing nest boxes and maternity stuff there. And I've also got a number of samples from Northern Territory and Mornington Sanctuary, courtesy of the Australian Wildlife Conservancy. So I've got these 5 populations that basically constitute the biggest population of Gouldians. Getting Queensland samples is extraordinarily difficult. So these people, as they were going out and surveying them, doing whatever they do, they also take blood samples. And so my job is then to take these blood samples and make DNA come out of it. So unlike human DNA, avian blood, sorry, is enucleated, so there's a lot more DNA per unit of blood in a bird than there is in humans and mammals. And so we can take some blood, and they've got all the blood cells have got chromosomes which all have our ATGC's genetic code in there. And this is a universal code, and this is what I am interested in. And so one of the genetic markers that I will be using to discriminate between individuals are something called microsatellites. So everyone has these. So basically they just represent a repeated motif of letters, and they are extremely variable between individuals. They have a very high mutation rate. So this unit can mutate in segments. So in one individual we might see 2 segments, in another individual we might see 3, in another individual we might see 5. And so this is very, very good if we use lots of these markers for distinguishing individuals. So these are sometimes the sorts of things that you might use in paternity analysis, establishing who the father is, in crime scene investigation, but it's also really, really good at looking at population structure and all that sort of thing as well. So basically, the majority of my PhD has been pipetting things into wells and getting RSI. And so this is what the microsatellites come out as, and I sit on the computer scoring these things, telling them what it is. And then I also look at mitochondrial DNA, which is separate from the nucleus, and I have a DNA sequence rather than these weighted things based on the number of repeats. And so those are the two kinds of data that I'm working with, just in case you care. So anyway, what we might predict about the Gouldians, as I said, we've got this quite fragmented range. And so we're interested in is their genetic structure in the Gouldian finch. So I'm going to present to you the results from a particular kind of analysis that I think is really quite visually and easy to understand. So this is the results from a human population study. So basically, if we don't have a lot of genetic interchange between populations, they are going to become more and more different. And using this analysis, basically what we see is that these are represented by these coloured blocks, so they're genetic clusters. And so all of these individuals, in Central Asia are more closely related to each other than Russians and Basque and French people. And so it's sort of found these different genetic clusters based on the genetic similarity from these sorts of markers that I'm using. And so these can also come out based on habitat structure or elements like big patches of forest or whatever. But I find that there's absolutely no structure in the Gouldian finch. So all individuals, no matter how far east or west, they are all one big population according to these methods. So this is not something that we expected to find, and this is something that we also see using the mitochondrial DNA as well. So this answers the first two questions of my PhD. So it seems that we have one big population operating here, and that these populations are very connected. I mean, we're one big population. So this has consequences for these survey methods. Basically, even though we can say that they're not enough, we can't say that they're completely wrong, but we can say that definitely we need to think about this a little bit more. Is that a question?
Yeah, I just wonder if you confirm the theory with a known diverse sample of two of the same species.
Sorry?
Whether you've proven that theory with two of the same birds that you know have diversity.
That have diversity?
Genetic diversity. You're trying to see if the Gouldian finch, there was any diversity in that sample. I guess, had you proven that way with a separate species to prove that does identify the diversity?
I'm not entirely sure I understand.
That's what, sorry, I don't mean
question.
That.
Sorry.
I guess it's like, what would you expect those results to look like if there was diversity?
If there was diversity.
Yeah, and how do you know what they look like?
Well, I mean, you would expect something
human.
that, oh sorry, wrong direction. So something like this, you might expect, maybe the Northern Territory populations are clustered in one position, and then, Mornington birds are quite different. Or you might see a gradient, for example, that maybe, even though there might be all be one sort of a population, the birds in the Far East are just gradually, less and less, more and more different as you go further west. But you don't see anything like that.
But if they were separated, how many generations do you think they have to be separated before they show up like that?
That's a tough question.
Maybe you haven't had enough time yet for it to show up.
Yeah, so
It's
basically,
possible.
I think Peri got museum examples, so you need a really large tentacle scale to be able to find that out.
Yeah.
Like if you have birds from, say, within the last 10 years, you're probably not going to pick up any sort of structural separation, which I will show in my talk.
True, but so just on that, so we're predicting that this separation has been going on for at least 50, 100 years kind of thing, and so this is perhaps the timescale we can pick these sorts of things up, and it has been picked up in the past, and we have lots of generations to go through.
The Gouldians, the majority of the Gouldians are down further than where you are. They're down around the Ord River in big numbers. Bigger than they've been for the last 20 years. Up at Go, you'll find flying around flocks of 400. West of Cairns, the federal government's put 100,000 in now for the Aboriginals to look after some.
Okay.
John,
that's good that they're getting some in Queensland.
John Griffith up there, he's dealt with the Aboriginals to look after all these birds. And there's also a colony on here where they're going to let them all go on the lake there. They found a big colony of Gouldians there as well.
In Queensland?
Yep.
Okay, well, we haven't been involved in that, so
I've got to say, they pick 10 pairs of birds in sitting out breeders and just see what their genetics are. Would they be similar, you reckon, do you or not?
To the wild birds?
Yeah.
No, I've actually, this is another chapter of my thesis, is that actually the captive birds are quite different genetically from the wild birds. Perhaps that's part of why they didn't do so well in Ariba in the first place. Yes, but they were captive re-releases, those. So.
Probably out there, that's why these
Sorry?
you've been for a river. You know, there's wetlands there, there's nothing for them to leak, it's overrun with cattle.
Well, that probably doesn't help either.
I've been to the river, the Blackford's still there.
Yeah,
Blackford, but yeah, still, the wetlands itself is still pretty good habitat for finches.
How long ago?
Last year.
That was it, yeah, 20 years ago. They'll let go up there.
Oh, yeah, the Gouldians, they're still releasing them there, they're still keeping them out there.
That's 20 years ago.
Yeah, but they're like none in the field. As soon as the bees get eaten by the outside.
In 1983, on the Ord River, they killed cotton farmers, killed 10,000.
Yeah, and that's
it was reported to the national parks over there. They look right up again. It's better than ever.
Yeah, so that's part of the issue, is that we can witness these very large population sizes, but this doesn't reflect the genetic population size. So we can have numbers of 10,000 maybe in 1 year, but then the next year they can crash. They can be virtually nothing. And that is more representative of the actual genetic population size, is that tiny population. So even though the next year we might have really, really, really high survivorship, the genetic diversity is extremely low in a very large population. So these numbers can be deceiving. And so that's part of why I'm doing this work. Okay. One thing I haven't answered yet is how big this population actually is. And I know I said that's a really important thing, but it can be quite difficult to estimate that properly. So that's what I'm working on today, kind of thing. So which is why I can't give you a good answer to those questions about how effective individual it is in the population, whatever. And I'll be using newer genomic technologies to answer some of these questions as well, which will give us more statistical power. And I've also got birds from museum samples. So we're collaborating with the Adelaide Centre of Ancient DNA, looking at birds before the decline to look at patterns of connectivity and population size before this massive supposed population crash, and how that compares with patterns now. And so this will be the really strong test of how much the Gouldian finch is really in trouble. Because if the Gouldian finch was already, had a small population size and was already kind of, whatever beforehand, then, we haven't done anything wrong, we can't, this is just the way they are, right? So this gives us a lot more information about how much they've actually been fragmented and what sort of things we might need to do.
It's mainly in the territory, I think, that's happening. Mainly in the Northern Territory, in Western Australia, they're in nearly played proportions again.
Yeah.
What did wipe out, the Department of Agriculture sprayed poison bird feed out of a plane all over one area and wiped a lot of birds out.
That's not
for a
right.
rat plague that was up there, and I reported to the authorities in Western Australia in 1983 when that happened.
Okay.
But dead birds everywhere.
Yeah, that's
But
unfortunate.
they built up a hell of a lot now.
Yeah, I believe it. I mean, that's what we've been observing, and that's why they've been delisted.
So they're pretty sick now.
So the new technologies that I'm using, they have much more statistical power. We've got, so the markers that I was telling you about before, they had 10 markers. I have 23,000 markers. And with this, I can use many fewer samples to get to answer the same question because I've got more markers. So I've actually got Queensland populations now in my data set, which is something. And preliminary results suggest that these are the same population as well. But again, you know, they could have been isolated quite recently. But genetically, they are similar. And this means that actually we could translocate birds from the west to the east to help build up those Queensland populations. And another question I'll be looking at is using these technologies. How genetically different are these morphs? So these technologies have already been used to identify the gene that is responsible for switching between red and black. And I will be using this to look at more about patterns of selection and what makes the different nuances in behaviour between the red and the black birds in the wild. And this is just a plot from my traditional data set using 10 markers. We see that there's no difference, but we can have very fine scale differences between the two different polymorphs or two regions based on selection on one particular trait, and that was just meant to illustrate that point. So Gouldian finches are, according to my data, genetically homogeneous across their range, which is a good thing. But it also means that these survey methods, in contingency with not really knowing the genetic population size, may not be necessarily the most effective way of estimating the population. But there's still more work to do, as I have outlined. And thank you to everyone who's been involved in the project.